Okay, so my doctoral thesis proposal is the discovery of novel small molecule SOS modulators and their implications in cancer drug discovery. First, I'd like to thank Dr. Wu Li for being my research advisor and inheriting me to finish out my PhD journey. I'd also like to thank my academic advisor, Dr. Windsor, for all of your help so far. Also, Dr. Tolius for inheriting me in the beginning of my PhD journey and also for being on my committee. And also Dr. Sharma and Dr. Wong for accepting the place in being a part of my committee. I look forward to all of your expertise in helping me in the future. So the first thing we should establish with SOS and KRAS is the entire MAP kinase pathway, which is shown in the figure on the right, and its implications in cancer research. So the MAP kinase pathway, the mitogen activated protein kinase cascade, is one of the most prevalent proliferation pathways, and it's present in every eukaryotic cell. So to go over how it's activated, we first have an extracellular growth factor, which is then bound to an extracellular growth factor receptor, the EGFR, which then works to activate the receptor by causing a dimerization and thus so subsequent autophosphorylation. This autophosphorylation leads to the binding of adapter proteins that can later recruit other important key effectors in the MAP kinase pathway. So one of those key adapter proteins, you have GREB2, which upon the binding to the intracellular side of the growth factor receptor, you have the recruitment of SOS, which is one of our proteins in question in this research. So upon SOS recruitment, that allows SOS to be in close proximity to already membrane localized RAS GDP, which that represents RAS in its off state, which I'll talk about more later. So upon activation or upon a binding of SOS to GREB2 and binding of SOS to RAS GDP, this allows SOS to enzymatically facilitate or catalyze the exchange from RAS guanine diphosphate GDP to the guanine triphosphate GTP, which represents RAS's on state. And upon RAS activation in its GTP state, it allows the rest of the downstream effect, which is binding to a RAF protein, and then the protein kinase cascade, which is MEK, ERK, and then the activation of transcription factors, which lead to increased gene transcription, cell proliferation, and ultimately cell growth. Now, this pathway is is very regulated, but in cancer, it can become dysregulated by certain mutations along the way. And the major mutations of the MAP kinase pathway include certain RAS mutations, those are listed here in this bullet point, RAS G12C and G12D, and also RAF V600E, which is an important mutation. These have been clinically studied. So to understand RAS-SOS interface or the binding partners as an important therapeutic target in cancer research, we first need to establish how prevalent they are individually or in that complex. So RAS mutants in general are prevalent in 30% of all human cancers, with the following breakdown in pancreatic, colorectal, breast, and lung cancer. So it's a pretty important target in cancer drug discovery. So to reiterate, RAS acts as kind of like a molecular switch, switching from its off-state guanine diphosphate, which is inactive, then being catalyzed to the nucleotide exchange is then catalyzed by a GEF. GEF stands for guanine nucleotide exchange factor, and our GEF in question is the SOS1 protein, which then binds to RAS, opens the RAS binding pocket, facilitates the exchange or the incorporation of GTP for that GDP. The GDP then flows out. Upon RAS GTP binding, this then activates RAS and allows it to bind to its downstream effectors. RAS is also turned off by the GAP protein, which is the GTPase activating proteins, NF1, but that's the negative feedback mechanism. So the idea here is we want to discover novel compounds that can interfere, either inhibit or hyperactivate or somehow modulate this interaction between RAS and SOS. And one of those ways would be the disruption of the interface between the two proteins, RAS and SOS, which would lead to a slower RAS activation, so a slower nucleotide exchange, which is the ultimate goal of a classical inhibitor. And then another approach is to hyperactivate this pathway by forming kind of like a molecular glue or somehow further facilitating the interaction between RAS and SOS to lead to cellular senescence that can happen in cancer mutants. So that is our aim, to discover and also to synthesize these novel small molecules that can modulate this interaction. To cover an important concept with the hyperactivation is cellular senescence. So studies have shown in the past that if you have a cancer cell line that has multiple oncogenic mutants, such as an EGFR and a KRAS mutant, it's actually shown to be deleterious to cellular responses and cellular growth, meaning it can cause cell death and what we call cellular senescence, which is the permanent arrest of the cell cycle because of a hyperactivation or just too many oncogenic mutants happening at the same time. So our idea with this project is to somehow synthesize small molecules that can mimic the secondary mutation. So if you have a already KRAS implicated cancer cell line, you can have a SOS activator or KRAS SOS activator that can further push this pathway to its extreme of activation, leading to this same response of decreased cell viability and cell death. And this principle was exhibited by Dr. Stephen Fesick's cell viability data, which is here on the left hand, on the left helicell, you have helicells and you have CHL1 wild type cells. And what you have here is Fesick's compound 4, FITC4, shown in the red. And you have in the black, you have a weaker compound, which is FITC5. Now, in the cell viability data, you have on the y-axis, you have the relative viability from 0 to 100, and on the x-axis, you have increase in concentration. So upon dose dependence, you see a decrease in viability with compound 4 at around IC50 being 44 micromolar in HeLa cells and 20 micromolar in the CHL1 cells. Whereas for compound 5, it's a lot weaker. The IC50 is in the actually hundreds of micromolar, I believe. So the idea here is that after incubation with compound, you can actually create, with an activator compound, you can actually create a situation where the cell goes into its senescent state and stops growing and eventually would die. So to further look at this interface and to see if there is a possible druggable binding pocket, you have the X-ray structure. So this is HRAS-SOS X-ray structure, the front view shown on the left and the side view shown on the right. Now you see in HRAS is in gray and SOS is in purple, you see this large binding cavity either along RAS and even possible binding cavities along SOS and along the interface in general. And this ligand is the guanine diphosphate, which represents KRAS in its off state, which that's the initial state that SOS would bind to it in order to facilitate the exchange of the guanine diphosphate to the guanine triphosphate. So the idea that this interface has potential binding pockets and along the active site of KRAS is very promising because it's relatively close, and you can somehow, if you bind a small molecule, you can facilitate some kind of interaction with the binding pocket, opening it more or closing it more to regulate the exchange of guanine diphosphate to guanine triphosphate. So this was interesting. This was an interesting approach and an interesting binding pocket. So what we've done so far is to first computationally dock our in-house compounds, which is a great place to start. That's our CHI compound library. And this was headed up by our computational chemist, Michael Sabio, who has successfully docked over 700 small molecule compounds from this in-house CHI library against the HRAS-SOS interface. Now, at first, this method was done in St. Louis, where they outsourced to supercomputers, which can bind in every possible iteration our small molecule compounds to every pocket along this, or every possible binding pocket along the SOS-RAS interface. So that would take a lot more power than a simple workstation, which is why we had to outsource it. And upon finding out that data that we have a potential binding pocket which has a low or as a high energetic favorability, we could then use that as our model to go forward with our future studies. And out of this screen, we have over 100 compounds that were selected as potential KRAS-SOS binding modulators. or just binders in general, which is a very high hit rate, but it's not really the amount of hits. It's just to narrow down our search to, and it really does a good job of narrowing down our search to find a potential activator or inhibitor. So the next step here was, now that we have this computational potential with these compounds, is to then dock them, or then to put them into a biochemical assay, which I'm going to talk about now, which is my fluorescent nucleotide exchange assay. Now, this is a biochemical assay done in solution and uses fluorescence to measure the exchange rate from the guanine diphosphate GDP to the guanine triphosphate, or the fluorescently labeled guanine triphosphate GTP. So the way this works is I first incubate SOS with Ras to allow a Ras-SOS complex to form. Then the compound, whatever compound it may be, an activator or an inhibitor or a compound that does nothing, is then incubated with that complex, allowing it to bind to either SOS, to KRAS, or to the interface if it does bind. Upon that, then BODIPI GTP, which is the fluorescent or fluorescein labeled guanine triphosphate, is then added to the mixture. This initiates the reaction, because as soon as the guanine triphosphate is in the presence of SOS, that is when SOS will begin to turn over its enzymatic activity and allow BODIPI GTP to be incorporated into Ras, and then also facilitating the exchange of guanine diphosphate out of Ras. So upon the incorporation of BODIPI GTP, we have an increased fluorescence reading due to the proximity of the fluorophore labeled guanine triphosphate and KRAS. So the idea here is that we can detect this, we can very finely detect this nucleotide exchange rate, and we can model it in terms of a rate constant or do certain methods of analysis. And in order to identify an inhibitor, there are possible modalities for an inhibitor, the way an inhibitor can be characterized. The first is by a direct binding to the KRAS-SOS interface, causing an inhibition or a slowing down of the KRAS-GTP exchange rate, which would be amazing if we can find a compound that does that. Also favorable would be binding to either KRAS or SOS that initiates a conformational change on either protein that weakens the interaction between KRAS and SOS. That's also very favorable and would lead to discovery of a novel inhibitor. Another way this can happen is the down regulation of the exchange rate will be by binding nonspecifically to either protein that would lead to either aggregation of the compound or of the protein, or some kind of precipitation or nonspecific effect that can decrease the exchange rate. This is not what we'd like to see. And then for an activator, something that would hyperactivate or increase the nucleotide exchange rate, this action we think would be as a molecular glue, meaning whatever compound, wherever the compound binds on either SOS or KRAS, it would facilitate the or create more opportunities for interaction of the other protein that would favor the protein-protein interaction between KRAS and SOS. Another way this can happen is by binding to an adjacent binding pocket on either SOS or KRAS, which can translate favorable conformational changes to the complex, which is another modality for activation. So in a cellular context, what we are measuring is when you hyperactivate SOS, leading to an increase in exchange rate, we would see an increase in fluorescence, which then leads to increased cell proliferation, theoretically, and tumor growth once we put it into a cell line. And also, if this gets hyperactivated more than the threshold allows, the cellular senescence mechanism will occur to our already cancer cell lines, which would have another mutation, let's say a Ras mutation, in addition to this SOS, this artificial facilitation of the SOS-KRAS interaction. Inhibiting SOS would decrease the fluorescence and overall lead to a decreased cell proliferation and tumor growth in our cell lines, which is favorable for classical inhibition. So the next thing is to establish this assay as working and is working properly. So we have a control for that. Now we have a commercial inhibitor, the KPEP2D, which was titrated in duplicates, and the literature IC50 via SPR is 1 micromolar. And we had this dilute, and this is what the data would look like for our normal guanine nucleotide exchange assay. So on the y-axis, you have, this isn't normalized, you have the relative fluorescence units, RFU, and on the x-axis you have time. So over time, you see that in the apo condition, meaning with no inhibitor, you have the maximum level of exchange rate, which the slope dictates the nucleotide exchange rate. So this is your maximum. And then as you titrate an inhibitor over a concentration gradient, you have a decrease in nucleotide exchange rate. And at 200 micromolar, which is shown in the blue, or I think in the blue, you have a complete basal level of really complete inhibition. It's almost a flat line. So this shows that this assay does, in fact, correctly measure inhibition or even activation of the nucleotide exchange rate. So these exchange rates, or even can be plotted as percent inhibition, and an IC50 can be determined, which was determined at 1.78 micromolar, which is very similar to our literature IC50 of 1 micromolar. So we know that this assay is valid for this purpose. So let's take a look at that was an inhibitor. Let's take a look at the kind of data we would expect for an activator for this fluorescent nucleotide exchange assay. Now we have the same kind of plot where on the y-axis you have the relative fluorescence over time. And you see that in the gray, you have the apo condition, meaning the SOS catalyzed nucleotide exchange, which shows some level of, or some rate. Then you have an activator, which is shown in the green, and duplicates are overlaying each other. This is one of our best activators, CHI47. At 100 micromolar, you see an increase in that exchange rate. So the fluorescence increases at a larger rate. And as a negative control, we have our cold, meaning unlabeled guanine triphosphate, which would be incorporated and quench the BODIPI fluorescent GTP signal once it gets incorporated more. And that would lead to a flatter signal because not as much fluorescent nucleotide is incorporated into the KRAS molecule. So this entire SAR screen, or this entire initial screen, was performed over 2 semesters with over 200 compounds, and an SAR was done, extensive SAR, on the compounds that we could buy commercially to figure out something, to get some kind of modulator, either an activator or an inhibitor. And then hits were characterized using a 3-phase analysis that's outlined on this slide. So just looking at the graphs themselves is not enough. We have to figure out some kind of quantitative analysis for interpreting this data. So the first one was obviously the visual analysis to see if visually our compound increased or decreased in nucleotide exchange rate in comparison to the apo condition. And on the left here, you have CHI113, which the compound shows to completely overlay the apo condition, which shows there's no effect on the nucleotide exchange rate at all. And then in comparison to on the right, you have CHI47, which shows a pretty big increase in nucleotide exchange rate. So that's a hyperactivation of that rate. The second is an endpoint percent inhibition analysis, where the RFU meaning the fluorescence units at 500 seconds or even 1500 seconds were plotted against concentration. So, and then that you can figure out an IC50 for a certain compound, or you can figure out a percent inhibition in comparison to the apo condition. So for example, if you look at this activator at 500 seconds around here, you see that this is maybe 30%, this is maybe 55%. So that difference is almost a 2-fold difference, which shows that you would have some kind of it would be a hit in terms of the endpoint percent inhibition analysis. And my favorite analysis, which is in my opinion the most quantitative and real, is the rate constant determination by PRISM. So these curves, these exchange rates can be plotted in PRISM with a 2-phase exponential decay. And the rate constant for the K fast, which would be the first rate, would then be compared to the apo condition, and exchange rates can be determined, and their percent deviation from the apo condition could be determined to characterize hits. So the first round of SAR was done with just 27 compounds. And these compounds were previously identified through computational docking by Michael Sabio, and they were biochemically tested by thermal shift assays by one of our undergraduates in 2017, I believe, 2018, Natalie Correa. And out of these compounds, there were several weak activator hits, but none of these were pursued for in favor of the other hits that we figured out. Now, additionally, there were 72 compounds from the CHI library that were identified from more computational docking campaigns. And out of these compounds, we do have several hits. We have a couple of activators that are shown in either weak or strong, shown in green, and we have one inhibitor that's shown in red. This was the only inhibitor, CHI 172. So naturally, we want to pursue this inhibitor to see if it is a novel KRAS SOS binding partner that can create inhibition or specific inhibition. So to do that, we had to do orthogonal studies. And the IC50 was very difficult to determine because it wasn't a weak inhibitor. So it didn't really show a large enough of a window in nucleotide exchange rates to be able to plot an IC50. So orthogonal studies had to be done. And the first was the thermal shift assay, which I'll get into more detail when we go over our activators with thermal shift assays. And basically, this was inconclusive. So the thermal shift assay would measure the binding of a compound to a protein by the melting of that protein. And then if there was a TM change, meaning a point of inflection change of the melting point of that protein, it would suggest a binding event. So that was inconclusive with this 172 potential inhibitor. And then another orthogonal assay was the same Bodipi nucleotide exchange assay, but this was in the absence of SOS. And the reason why I did this was to measure the intrinsic nucleotide exchange rate. That's SOS not catalyzed. So the thought here was, I did read a paper that was similar to this, and they did the same thing. And they saw that if you have a novel inhibitor, or since the intrinsic nucleotide exchange rate is so low compared to the SOS catalyzed exchange rate, then if you have an inhibitor or some kind of compound that greatly affects the intrinsic exchange rate, then it suggested that the only way to do that would be by messing with or binding to an important part of the KRAS binding pocket that can potentially be nonspecific. And by this, I mean the magnesium cofactor, which is important for the guanine nucleotide binding to KRAS's active site, or it could be interactions with the protein that are greatly nonspecific and cause precipitation, or some kind of binding to the guanine nucleotide itself, which can inhibit the binding of the guanine nucleotide to KRAS. So that's a pretty weak argument that they made, but my data actually suggested that it was maybe true or something, they were onto something. Because as you can see in this SOS absent, or really the intrinsic nucleotide exchange rate assay, the apo condition shown here and at 200 micromolar shown here, it showed a pretty big decrease in activation or a pretty big inhibition. So that made me think that, all right, maybe this isn't a specific inhibitor. But there was more to test to elucidate that. So the next thing what to do, since that was kind of a weak argument, is to take more CHI 172 like analogs from our in-house library and screen them for potential SAR. And out of those compounds, none of them showed any kind of visual analysis increase or decrease in nucleotide exchange. So I didn't even bother doing the quantitative analysis since the data was so clear cut that they were overlapping. So to me, that showed that for some reason, something about 172 showed a non-specific effect on KRAS SOS complex, or non-specific inhibition, which is not the best, is not what we want to see. So this shifted our primary research focus to the discovery and development of the established SOS activators that were discovered in our screen. And there were 3 of them. And these 3 were 47, 159, and 167, which before I even looked at the structures, they showed about the same EC50s. But then once I looked at the structures, I saw that makes sense because they are all basically the same structure. They have the same core. They have the same primary amine group that's protonated at physiological pH. And they have the same benzyl group with different substituents. And the fluorine is in a different spot. In 47, it's in the para position. In 159, it's in the meta position. And in 167, it's a methyl. So they're very similar compounds of the same class. This was very good to see because now we can pursue this class as a whole and further develop our SAR. And these compounds were shown to not influence KRAS intrinsic nucleotide exchange. So that suggested that it was some interaction with SOS or KRAS or the interface itself that was specific to KRAS SOS, which caused an increase in nucleotide exchange rate. So that's very important. And also their EC50s were about the same, 20 to 30 micromolar, which is a good place to start. And something else that was interesting is that this core, upon further study, is caffeine. So this is a novel caffeine core, is what we refer to it as now. And it's important. It's a novel thing to study. It's very interesting. If we can develop a caffeine core that has some KRAS SOS inhibit or activation activity, which can lead to senescence, that would be a big story. So the 5th and 6th round of SAR and further studying this core and what can further activate or inhibit it or activate it, we ordered more compounds from different chemical vendors and we basically exhausted all of our database or all of our chemical databases to figure out to extend the SAR as much as possible before synthesis is developed. So this is the core. We have position, which we call R1, which is this benzyl ring. And if you look at 47 on the right, which is the benzyl, and then R2, which is this primary amine group. So we then classified the compounds that were ordered, and there were about overall maybe 40 to 60 of them. And we classified them as class 1, in which the R1 region, which is the benzyl group, is a variable domain, and R2 is a fixed primary amine group. So it has this protonated amine at the end. The class 2 was where R1 was fixed as some kind of benzyl group with a variable substituent, but the important thing was to have that benzyl there. And then R2 was variable. And here's what we figured out. So we do have more hits, but if we look at structures on the right side of all the hits, we can take a look, and we'll look at them quickly one by one. As you see in the R2 region, where you have the amine, you have primary amine, amine, amine. Most of them, except for 859, have that protonatable amine group in that position. So most of these class 2 compounds, or sorry, these class 1 compounds, they showed activation, some level of activation, whether that be weak or strong. So whereas the class 2 compounds, which did not have this amine, none of them really showed activity. So that leads me to believe that this primary amine is important for some kind of interaction it's making with either SOS or KRAS. Up until this point, what we know now, it's an SOS modulator, but up until this point, we didn't know if it bound to KRAS or SOS. So that's interesting to note that this is important. So this SAR, we're developing it, and we saw that the different benzyl substituents were also important, and the position mattered. So the power position and the meta position were favored, as opposed to the ortho position right here, which it showed activation, but not as much. So another kind of side thing we had to do was to test these SOS activators in the presence of the mutants, into KRAS mutants, because what's the point of saying that we're going to cause a cellular senescence if we're just testing on KRAS wild type? We need to also use the mutants, and which is in the cell lines that we test eventually in the cell assays, the mutants are going to be the most important. And we see that in G12C, and also we did a double mutant G12C T58A, that the IC50s for our main 3 activators were shown to be about the same in the 20 to 30 micromolar range. So and we see that same thing for the double mutant G12C T58A. We see they're in about the same range, which is good. And it shows us that that also leads us to believe that these compounds may have more of an effect on SOS than they do on KRAS, because the mutants do behave differently, but the SOS in each case was the same. That leads us into actually determining, or a better assay to determine which of the binding partners, either SOS or KRAS, that these compounds attack, or these compounds bind to. And that was by the fluorescent-based thermal shift assay. So the way this assay was done is that if we look on the left here, we have fluorescence on the y-axis and the temperature gradient on the x-axis, and we have a protein, basically a protein melting curve. And this protein melting curve fluorescence is dictated by the environmentally sensitive cipro-orange dye, which is incubated with the protein. Upon melting, or upon increasing the temperature of the protein cipro-orange complex, you have an unfolding of the protein. which this cypral orange would then bind to the exposed hydrophobic regions of the unfolded protein, since cypral orange is very hydrophobic in nature, and it's environmentally sensitive to binding to other hydrophobic regions. And then from that, upon the protein unfolding, we have an increase in fluorescence, and then the fluorescence decreases later on for several reasons. But the important part is that the inflection of this curve can be denoted as our TM, or our melting temperature of the protein. And the idea is that if we incubate this protein with a compound that binds to it, it will either increase or even can decrease, or the idea is to just change the melting temperature. And this assay is very sensitive. Even something as low as 1 or 2 degree change is considered to be significant. So what we did was to incubate both KRAS and SOS to mimic the complex, and then we then put our compounds in there, either our APO condition, which is no compound shown in red, and our 200 micromolar of CHI47 activator shown in blue. And we have 2 distinct TMs, so 2 distinct melting temperatures, since this mixture has 2 proteins in it. So the SOS melting temperature TM1 is shown here as the first inflection point, and then the KRAS TM2 is shown as the second inflection point. So luckily, in some cases, you have proteins that have a kind of overlapping TM. In this case, in that case, this wouldn't be able to be done. It would be one big broad curve. But luckily, their TMs were far enough apart, being SOS is 44 and KRAS is about 52 to 53. Then we can do this assay and see specific binding for TM1 or TM2 shifts. So let's look at that data for CHI47. If we analyze TM1, so TM is on the y-axis and the concentration is on the x-axis, we can see that at a very low concentration, your TM is about 44. And then as you increase the concentration, you get a TM is about 44.6 to 44.8. So that's a very small shift, but it is a small compound, and it's also affecting a very large protein. SOS is huge. So having a small shift like that, and this shift was reproducible. This trend was reproducible, which is, I didn't believe it at first. It was only a little bit more than half a degree, but I reproduced this multiple times and saw that SOS TM did have a concentration, a dose dependence with incorporation of the activator, as opposed to the KRAS TM, which the KRAS TM was kind of all over the place, didn't really show any kind of trend. So it had a general, no matter how high the concentration of the activator was, it didn't really show a TM shift in one way or the other. So that leads us to believe that SOS is the compound's binding partner. So we know that. Now we can do another orthogonal assay, which is SPR. So SPR, a surface plasmon resonance, is a label-free biochemical technique to measure either protein-protein interactions or protein small molecule binding interactions. In our case, it would be protein small molecule. And these curves are denoted by an on rate, which in this data right here is a control data for CA2, carbonic anhydrase 2, and its substrate furosemide. So they would bind with about 0.5 to 0.7 micromolar affinity. And as you can see, at first, when you have the start of the injection, the compound would then bind. There's an on rate. There's a saturation point where the compound can't bind anymore. And then once you stop the injection, the compound begins to flow off with a measurable off rate. This measurable on rate and off rate could be used to detect or to measure and calculate a TM for whatever substrate or ligand or analyte we're talking about. And this TM, and either the curves can be kinetically fitted, and then a point study could be done.
Patrick?
Yes.
Patrick, you said TM, you mean KD.
Oh, KD, I'm sorry. So yeah, KD. So K off divided by K on would be the KD. And then if you take endpoint analysis, meaning just pick a point of either of these curves, either of these concentrations, you could then plot them in a point study to also figure out an apparent KD. And these are about similar for the CA2 furosemide experiment, which is a control experiment, and it's very well documented. So this experiment was done with our activators. And here is CHI-47 shown in the left. And on the right, we have the point study, which is just plotting these saturation points with respect to concentration. And on the left, we have a very quick on rate followed by a saturation. And it's a dose-dependent saturation, which is good, which means it's probably specific to SOS. And we have a very quick off rate. Now, this very quick off rate is not really calculatable. Same with the on rate. And this is indicative of a weak inhibitor or a weak binder. So that's something we'd expect to see at a binder that is in the tens of micromolar affinity. So that makes sense. And we were pretty happy to see that. And if we did plot that activity, we saw that the apparent KD was about 50 micromolar, pretty similar to what we were getting in our biochemical assays. And then this same assay was performed with the rest of the activators. And we see a mixture of activity. Most of them confirmed activation and around the KDs that were the same or very similar KDs as the EC50 shown in the biochemical assay before, the nucleotide exchange assay. And with only one exception of 159, didn't show good binding, I'll put it that way, because it showed some kind of response as opposed to something like we had negative controls in this assay. CHI 806 was considered a negative control, and that data was completely flatlined. So the fact that 159 showed some kind of increase, even though it's not concentration dependent, shows that maybe something's going on, but maybe it's very weak. And the other one showed pretty good activity, pretty robust and reproducible activity in comparison to their activity on the biochemical assay. So then we need to take this data into context of where our compound is binding and exactly its mechanism of binding. So the Fesic lab at Vanderbilt University discovered similar compounds that they are their benzimidazole derived core that was incorporated into its synthesis and into an SAR campaign, and they have their best compound has an EC50 of 9 nanomolar. And that one's not shown here. They didn't do extensive crystal structures on it, but they show this also very potent inhibitor or activator, Fesic compound 44, which had a KD of 44 nanomolar. And here is the SOS binding pocket that they believe the compound binds to according to X-ray structure. Here, so you have very key interactions with the aspartic acid 887 and the glutamic acid 901, which is it's right here. And every other interaction that the compound observes is deep hydrophobic pocket binding. So you have that hydrophobic pocket, and you see this benzyl ring is kind of sticking deeper in that cavity. And if you look at the structure of Fesic compound 44 in comparison to our most potent activator, they're pretty similar. They have their bicyclic core, and they have the benzyl group with the substituents, and they have the primary amine, which is protonatable at physiological pH. So before we knew about Fesic's data, or before I read and looked over Fesic's data, excuse me, I have to sneeze. Sorry, I don't, sorry. So before looking at Fesic's data, we didn't know that it has this possible binding mechanism. And then looking at Fesic's data, everything clicked and everything made sense because we have a similar compound that has a novel core and that behaves, we think, in a similar manner of similar binding modality. So our job next is to optimize this caffeine core to maximize its potency. Also to optimize the core itself by addressing its weaknesses, since structurally, when we did the docking study, it did show some structural weaknesses. Those being the carbonyl groups had some kind of steric hindrance with the hydrophobic pocket around this area, which I'm showing with my mouse. And also a further objective is to begin to explore the SOS inhibitor scaffold, which was found by Baer in 2019. So it's a further look at in the 3D in a surface model of the KRAS SOS interface and the activator. You have HRAS that's in gray, and you have this is the interface, and then you have SOS in purple. You see that there's a very deep, you can't even see the benzyl group down here. It's in the deep hydrophobic pocket. And then you have the aspartic acid 887, which is over here. Then you have the glutamic acid 901, which is around here. And then you have other space-filling interactions. Most of the compound, besides for those two major interactions, are hydrophobic and space-filling. So we believe we have strong suggestions that our compound binds in the same area. So the future direction of this project is to synthesize, which is currently ongoing, synthesize structurally optimized compounds, basically taking our caffeine core, incorporating some of Fessek's ideas, and kind of optimizing it based on the data we have. Because no other compounds that can further our SAR efforts can be purchased, because we exhausted all of our resources or everything that's available. Then also these compounds would have to be validated by the biochemical and biophysical assays, and then put into our cell-based assays, which is going to be our cell proliferation assay against wild type and mutant cell lines to measure cell growth, our Western blots to see changes in phospho-ERK levels, and also our immunoprecipitation assay to detect the level of KRAS GTP level in response to the activator. So the synthesis, there's several compounds that we are going to synthesize, and here are 5 of them. And you have on the top here, you have Fessek compound 44, and then you have our activator, 826. So the first thing we want to do is optimize this core, because the more compelling research would be with a novel core, especially if it's caffeine. So we want to keep this caffeine core as long as possible and try to maximize the affinity. So the first compound, which I'm working on now, I guess I'll go to the next slide, is right here. And we have our caffeine core with basically, well, actually I should go back, with Dr. Fessek's substituents being the dimethylfluorobenzyl and the diazospiroheptane group. And then also we have this piperazine here that's shown to have an interaction with the glutamic acid 901. That's the major interaction that we're not capitalizing with our current activator, which I think once we have that attachment, it will greatly increase the potency, because looking at his SAR, it showed to do that. Once they got rid of this ring altogether or anything in this substituent area, then potency was decreased by a significant margin. So the synthesis, and here's two examples of the synthesis of compound 1 and compound 2. All the synthesis will be very similar, but they will have different starting material cores. So the first step, we would have to brominate the 5-membered ring, but we use NBS or we could use bromine. The recent way I did it was just using liquid bromine. And then we can do a series of nucleophilic substitutions to put our certain groups in there, whether they be our dimethylfluorobenzyl and our Bach-protected diazospiroheptane, which would add here. And then we can do another nucleophilic substitution of our substituent that belongs here, that the Fessek substituent that supposed to greatly increase activity. And then we can do debach deprotection to come up with our final compound. So these are just 2 examples of that. And this is in progress now. So the cell proliferation assay, once we develop these potentially, or once we validate them by NMR, by mass spec, and validate that they're the compounds, and also by the biochemical assays, we could then put them into our cell-based assays. The first cell-based assay would be the cell proliferation assay, which is Alamar blue, which represents and measures cell growth by detecting the reduction of the reduction of a dye that, upon reduction, changes color and then fluoresces at certain wavelengths. And this will be done with HeLa cells and NCI H727 cells, which have a G12V mutation. So what we expect is that the oncogenic cell lines will show little change in activation at low concentrations, because we don't imagine them to be nanomolar potent, because of our core isn't as optimized as we think it could be. But we do expect, and Fessek's group didn't see this either, even with their most optimized compounds. But we do expect to see at higher concentrations a decrease or a hindrance in cellular growth, leading to cell senescence or a downregulation of the MAP kinase pathway. And also, we expect wild type cell lines, which don't have KRAS mutations or MAP kinase mutations, to show increase in proliferation with the SOS activators. The next would be a Western blot analysis to analyze p-ERK levels. That p-ERK ERK gets phosphorylated downstream of SOS-KRAS. And we expect the weak activators to show a concentration-dependent increase in p-ERK levels until, even at high concentrations, maybe they will show some cellular senescence or a decrease or a stagnation in the p-ERK levels at these higher concentrations. Now, the data shown here on the bottom left is a weak activator of FESX, compound 55. And you see, if you look at p-ERK, as you increase concentration, the level of p-ERK increases in a dose-dependent manner. Whereas if you have compound 64, which is a stronger activator, you see that the level of p-ERK is increased in a concentration-dependent manner, but decreases as you get above 1 micromolar, which shows that at this level, this is the threshold at which the MAP kinase pathway starts to turn off in a mutant cell line. So that's due to cellular senescence and negative feedback loops. The next assay would be the Ras-GTP pull-down immunoprecipitation assay, in which we would incubate our cells with the, in this case, we would use HeLa cells, which have wild-type Ras. We would incubate them with our activators. And then we would take the lysate, we would incubate them with GST-RAF, which RAF, so the Ras binding domain of RAF. So RAF is Ras's downstream effector. So the idea is that only the on-state Ras, which is Ras-GTP, would bind to GST-RAF. Once that binding partner is established, we then put in the resin, the glutathione resin. The glutathione resin would bind the GST-RAF-RAS complex, which would then everything else could be washed out, and then we can elute using a eluting buffer. What would be left is the Ras-GTP, which could then be, we could take a Western blot of that to see the direct activation by the activators of SOS, which then immediately lead to Ras-GTP activation. So we can monitor that with this assay. So the conclusion of all of this research would be it has value in both the field as well as my educational pursuit in my knowledge and also in my doctorate. So in biochemically and cancer biological relevance, the first thing is that development of a novel caffeine core that influences MAP kinase activity can be a potential tool compound or even stimulate new avenues and new research into this binding pocket and see how it can further be optimized, or also potential binding pockets that are nearby. And since, remember, the X-ray structure shows it's pretty cavernous, so there may be more that are worth discovering. And especially the novel caffeine core is really the big punchline here. So also importance in my pursuit of knowledge in doctoral research and would be the kind of the full picture of the drug discovery campaign, which I have experience, I will have experience in every facet of it. That's starting from small molecule screens, biochemical, enzymatic, biophysical assays, data interpretation, analysis, also small molecule organic synthesis, which is important, computational modeling using the Schrodinger suite, which I'm pretty good with now, and then using this data to optimize compounds and then put them into cell-based assays such as Western blots, cell proliferation, immunoprecipitation assays to further validate my findings. So doing all of this I was very grateful that I can get experience and exposure to all these different techniques. And then the future of the Windsor lab, this work already has spawned certain research questions that can be pursued by future students, and that being the optimization of the caffeine core. Also exploring the SOS inhibitor and using our caffeine core to kind of mold it with a inhibitor that was already discovered by Bayer, but that's more theoretical, and also elucidating more information about SOS regulation, which is a very complex process, which in some it's not well understood to the exact specificity that it should be. So with that, I conclude, and I'd like to thank everybody for listening, and I ask any questions.
Thank you, Patrick. Will, how do you want to go forward?